Already a question.
Yes, yeah, okay. So I guess we're on now. So hello everybody, and hello Tom and Joanne. I'm going to share with you a couple of introducing slides. Just bear with me for a second. Good. So now officially, hello and welcome to our webinar on biotransformation. As you know, biotransformation and the biotransformation of hop compounds, either by yeast or added enzymes, is currently a really hot topic in the brewing industry. So in the last years, research on biotransformation became really a focus not only of institutes and universities, but also by hop and yeast producers. We at Lallemand Brewing, for instance, screened all our brewing strains for their beta-glucosidase and beta-lyase activity with different methods. So some of our results are presented in two best practice sheets that you can find on our website at lallemandbrewing.com. Besides the biotransformation of the activity of the yeast, we also worked on the development of a new enzyme formulation to enhance hop flavor and aroma in beer. This new product is called AB Vickers Aromazyme, and we just launched it this week, so it's now available to you. This is actually our second webinar on biotransformation. Our first webinar was with Laurent Degas from Nisius. And he covered the theory of biotransformation and the analysis of thiols and terpenes in wine and beer. If you missed this webinar, you can still watch Laurent's presentation on YouTube. I have the link here in this slide. And I'm sure these slides are available later on, so you can do that in patience, if you want. So this is now in the second webinar on biotransformation. We're focusing more on beta-glucosidase activity, and it's a bit more practical oriented than the previous one. And we're presenting pilot brews and field trials. So we're very happy to have Dr. Tom Shellhammer with us today to present some of his work and answer your questions. In the first part of Tom's presentation, he will present basic of hop biotransformation, and then in the second part followed by pilot brews, results with selected brewing strains and two different enzyme formulations, including the AB Vickers Aromazyme. Then my colleague, Joanne Montesel, will then present results from field trials with a Spanish brewery, also including these AB Vickers Aromazyme. Oops, sorry for that. So I'm sure most of you know Tom Shellhammer. But for those who don't, I will give you a little brief biography. So Dr. Shellhammer is the Northwestern Professor of Fermentation Science in the Department of Food Science and Technology at Oregon State University. He's an internationally recognized expert in hops chemistry, in brewing science, and his lab investigates hops, beer quality, and the origins of hop aroma and flavor in beer. He served as president of the ASBC in 2014 and 2015, and as the president of the District Northwest of the MBA in 2016 to 2018. He currently serves on the board of examiners for the Institute of Brewing and Distilling and the Brewers Association Quality Subcommittee. So welcome, Tom, and I will try to stop sharing now and give you the screen.
All right, thank you very much. Tobias, for that introduction, and exciting to be here. It's really fun to watch this tech stream rolling up on the side here with people from about like 4 or 5 different continents joining on this. So I think it's really cool how the upside of COVID is that we're all very comfortable with using technology that can bring people together rather quite easily. So as Tobias mentioned, the concept of biotransformation is something that is kind of forefront of a lot of people's attention right now, particularly hop biotransformation. And so that's what this talk is about. And as Tobias mentioned, we're going to talk a little bit about sort of the fundamentals, as I see them, of yeast biotransformation, and look at some case studies, some that we carried out in our lab. And then when I'm done, I'll hand the baton over to Jawan. He'll talk about some industrial-based case studies. So, the concept of biotransformation can span a whole variety of different areas. And I think probably an overly simplistic view of this is the yeast ability in an alcoholic fermentation to transform fermentable sugar into ethanol. And brewers are very keen at monitoring that. We follow fermentation curves. And while we don't necessarily follow ethanol production, we know that there's a simple stoichiometric relationship between the amount of sugar consumed versus the amount of ethanol that's produced. And depending upon whether you're a brewer, a distiller, a winemaker, you're very keen on paying attention to the fermentation performance. But that, I think, is an overly simplistic view, because what we're talking about with the biotransformation of hops is not just the conversion of one material into another, but a series of cascades of reactions that can influence and modify hop flavor. So in order to talk about hop flavor, I think it's helpful if we spend a little bit of time just talking about hops. And while they're certainly a source of bitterness, we're not really going to focus on bitterness in this lecture. We're going to talk about hop aroma. And if you understand where hop aroma comes from, then it makes more sense in terms of the biotransformation aspects of that. So here's a table of various things that you might find in hops. Not all of these are of value to the brewer, particularly the major fraction, the cellulose lignin fraction, is stuff that just passes through the system. But the part that we're interested in for today's talk is the hop oil fraction, which is actually relatively low in hops. It can be as less than 1% or so. But this essential oil fraction is volatile. It's made up of, well, we'll talk about the various compounds that it's made up of, but it's a very complex mixture. And the fact that it is volatile presents some interesting challenges to you as a brewer in terms of how you use hops and where you use hops. In the lab here, we use the volatility to actually capture the hops. We take hop samples and grind them up and put them in a flask and hydrodistill them, capture them in a receiver, and we can just look at actually how much oil is coming over from a certain amount of hops that have been put into this vessel. And then we can sample this oil mixture, add it to a solvent and shoot it on GC and find out what's actually in it from a compositional makeup. So hop scientists realize that there's a lot of complexity in hop oil, and we kind of distill this down, no pun intended, to some major classes of compounds. And Sharp and Laws published this back in the 80s, where they propose classifying hop oil in these 3 different compounds: the hydrocarbon fraction, an oxygenated fraction, and a sulfur fraction. And we'll spend some time going through each of these. Various chemists have been looking at hop oils, and a paper published by Graham Ayres and Jean-Pierre Dufour note that there's well over 1,000 different compounds in hops. So it's a very complex mixture. Not all of these are at levels that have a sensory influence, but the fact that there's such a broad diversity of compounds makes studying them somewhat of a challenge. But I think if we try to simplify this in terms of these different fractions, it helps. So if we start with the fraction that is the most prevalent in hop oils, the hydrocarbon fraction, and these numbers here, these are really gross generalizations. Some hop oils you can find as much as 85% of the oil compounds being made up of hydrocarbons. But the big players here are myrcene, humulene, caryophyllene, to a minor degree limonene. Myrcene is a monotropian. So you can see this is the compound here. It's a hydrocarbon. As such, it's not very polar. It doesn't really want to be in water or beer very much. And if it is there, its solubility limits are low. Myrcene smells very resinous. It's like green hop aroma. It's very characteristic of that myrcene smell. You also find it in things like carrots and bay, so it's found in other materials quite well. Humulene and caryophyllene, these are larger compounds. So these are sesquiterpenes, and their polarity is even lower than that of myrcene. Their solubility is considerably lower. If we look at that in terms of partition coefficients, their partition coefficients are these oil-water partition coefficients are huge. They really want to be in an oil phase. They really don't want to be in a liquid phase. And as such, they don't really contribute to hop aroma per se. And neither does myrcene, even though it's there in large amounts. You just don't find a lot of it in beer. And the levels that are there are often lower than detection thresholds. Now, some of the oxidation products of these can have an influence on beer flavor. And certainly when we talk about dry hopping, when you've got basically a suspension of hop oils, these then start contributing to hop aroma. But these middle two here are very woodsy, spicy kind of aromas. You find humulene in cannabis, you find it in thyme, in caryophyllene, in basil, and cloves. Limonene is citrusy, and so that's a very different smell of these guys. But in general, these compounds are woodsy, resinous, and spicy in aroma. If we look at the oxygenated fractions, these are the monotropians with oxygen in them. So linalool has just got an alcohol group in it. And the fact that we have some oxygen dramatically increases the polarity. It dramatically increases their, lowers their oil-water partition coefficient and increases their solubility in wort and beer. So you're going to find these at much higher levels, and they persist. They persist through boiling. They persist through hot side additions in the whirlpool. And these compounds, as a class, have a very different type of aromas. So linalool is very much a floral as well as fruity compound. In the U.S., if you as a child you ate Tutti Frutti Pebbles, there's a lot of linalool in that. So it's that fruity, floral kind of aroma. Geraniol is floral. Citronellol is floral. In the U.S., we use insect repellent, like in citronella candles. Nerol, you find that in lemongrass. It's also floral. So it's fruity and floral in this class. And geraniol acetate, which is an acetate ester of geraniol, you find that as well. And it's also a kind of that floral, fruity category. So it's a different class of compounds, different degrees of solubility and certainly polarity, and a different class of aromatics to them. And then the last group, which is the smallest group that you find, 1% or less in hops or hop oil, are the sulfur compounds. And the sulfur compounds are particularly important because they're very, very potent. Three compounds, these thiols that have received a lot of attention in the last, I would say, 10 to 15 years or so, particularly in the last 5 to 10 years, are 4-MMP, 3-MH, and 3-MHA. And we use those abbreviations because the names are a mouthful. So 4-mercaptose, 4-methylpentane, 2-one is hard to roll off the tongue. We just call it 4-MMP. And 4-MMP, it's got tropical smells. It's got black currant smell, depending upon its concentration. It's got kind of a cat pee-like aroma. And I've heard some people refer to this as the cat pee thiol. And the concentration of these in beer will dictate their aroma. And I have a funny story. I have a student in the lab, had a student, Dr. Sharp, and he was making 4-MMP stock solution. And he made this in the hood. He made it during the weekend. And when I came in the lab on Monday morning, the lab smelled like a zoo. I mean, it smelled like body odor. It smelled like a veterinary hospital. It was just so animal smelled. And he sort of sheepishly said, like, he had made these dilutions. And even though he was being as careful as he could, a little bit of it got into the lab. But what was interesting is that every day throughout that week, as the concentration got lower, the smells changed. And by the end of the week, the lab smelled like kind of like a tropical paradise, and away from that cat pee aroma. 3-MH and 3-MHA, the acetate ester, that smell very much tropical. They smell like passion fruit, guava. We find this in these compounds in grapes. We find them in Sauvignon Blanc. And in fact, a lot of the initial research was done by the grape community, the wine grape community. And a New Zealand Sauvignon Blanc has a fair amount of 3-MH and 3-MHA. In fact, a hop called Nelson Sauvin, it's got a lot of this functional thiol in it. Should point out, though, that these sulfur compounds, while they're present there in free form, there's even larger portions that are present in bound form. They're either bound to a range of different compounds, commonly cysteine and glutathione, and in their bound form, they're odorless. And part of this biotransformation talk that I'm going to get into is about releasing those compounds. So it's the relative combinations of these and the interplay of these components that influence different types of hop aroma. And they influence to different degrees. Our lab about 5 or 6 years ago was looking at aroma thresholds of these individual compounds in beer. And these numbers here represent those detection thresholds. And so some things to see is that compounds like beta-caryophyllene, we're closing in on 1 ppm levels that you would need to have in beer in order to be able to smell them. And the solubility limits are way, way below that. So it's often you don't see that as an aroma contributor. Well, linalool and geraniol, these are found at higher levels and can be found above these threshold levels. A thing that I should point out to you, though, is that look at the sulfur compound FORM-MP threshold. So it's like 5 orders of magnitude lower. It's 6 parts per trillion in comparison to hundreds of parts per billion on these other compounds. So a little goes a long way. And one of the challenges in terms of looking at sulfur compounds is that you don't find them at large amounts in hops. They're very potent. They're found at very, very low levels in beers as well. And so you need some pretty advanced instrumentation to be able to track those down and identify them when you're trying to hunt for things in the PPT level. All right, now let's talk about yeast hop interactions and move towards biotransformation. So if we think about like that first slide, kind of the basic interaction that you see between yeast and beer is the production of ethanol. But it's not just ethanol, we get ethanol and CO2. And so during fermentation, CO2 production can result in volatilization and stripping of hop compounds during fermentation. The yeast cell walls are very, they're very, I would say, active and that they can actually absorb a lot of the hop compounds onto the yeast surface itself. If we grow up yeast in the absence of hops and tasted it, okay, it tastes yeasty, maybe slightly bitter, but not terribly bitter, but you take yeast that's come out of a brewery and it's extremely bitter because of the surface being coated with isoalpha acids. There's also a fair amount of hop compounds that could absorb to the surface as well. And then we've got foam that is being produced during fermentation. And depending upon the nature of fermentation, we can actually get partitioning of hop compounds into that foam as well. We certainly see this with bitterness, but we can also have that happening with volatile fraction as well. And as fermentation progresses, you get ethanol production. This in turn changes the solubility of the beer itself. And that's one reason why dry hopping is kind of a cool approach, is that you've got some ethanol, the solubility of these compounds goes up, and so you can get more hop material into solution. I think the other thing that brewers have realized is that because there's no isomerization going on the cold side of the process, you're not really limited in terms of high ends of using hops. And so you see brewers that really want to create hop aroma put a lot of hops in during dry hopping. And in some cases, they're limited only just based upon the size of their vessels. Of course, with that comes tremendous beer losses, and it's not a terribly efficient process. You can also have aroma masking. I've noticed in some of the research that we've done involving yeast that are POF positive, that things like 4-vinyl glycol, I believe, have a kind of masking effect on some hop aromas. And then finally, we get to biotransformation, and that's kind of the point that I wanted to talk about. This slide down here is coming from a paper by King and Dickinson. It's definitely a good read if you're interested in biotransformation. And this basically is showing the loss of the sesquiterpenes during fermentation. And it's a loss due to these things here. We get some CO2 production as well as the absorption that is basically stripping these compounds out of solution over fermentation. There's a whole slew of different types of biotransformation of hop-derived compounds. And I'm going to talk about 4-ish classes of these. It's called 4. One is the hydrolysis of esters, or the production of esters by esterase enzymes in hops. We know that yeast produce esterase enzymes in yeast. We know that yeast produce a lot of esters. They can trans-esterify different hop compounds. They can break down hop compounds, and they can actually esterify hop compounds. And so This is a paper that was just published recently by Kiyoshi Takoi. He's a hop scientist at Sapporo. And what we've got across the bottom here are a range of different types of medium chain fatty acids. Some of these come from yeast. They come from the degradation products of yeast. These are the side chains of alpha acid. And the 3-methylbutyric acid just comes from the production of medium chain fatty acids by the yeast themselves. These guys at the bottom here smell like cheese. They smell like sweaty socks. They smell a little bit like baby vomit. So these medium chain fatty acids are not pleasant smelling compounds. But the yeast had the ability to actually esterify these, and the ethyl esters of these sneaky compounds smell citrusy or fruity. That's what ethyl isobutyrate smells like. Ethyl isovalerate can smell like pineapple, slightly like bananas and apple. And then the ethyl ester of the 2-methylbutyric acid smells like green papaya or strawberry, pineapple, or maybe even strawberry. So we see quite a transformation in flavor from something that would be unpleasant or off-putting into something that is actually quite pleasant. And the hops, yeast also have the ability to take esters that are found within hops and trans-esterify them. So things like from isobutyl isobutyrate into ethyl isobutyrate. And these compounds here, given that their esters are in that sort of fruity category, tropical as well as general fruity characteristics. And these are just examples of people you can look at in the literature who've studied these, and there are plenty of others. So this is the ester story and the ability of yeast to create esters out of hop products. The next story is monotropian alcohol isomerization, back to the paper by King and Dickinson. What is interesting, what they discovered, and what I think is interesting to see, is that a compound like geraniol, which is almost like one of the poster child of hop aroma from hops, that along with linalool, it is pretty dynamic during fermentation. And they looked at geraniol, and they saw a decrease in geraniol over time, but saw increases in beta-citronellol, increases in linalool, increases in alpha-terpineol with fermentation, despite the fact that the total terpenoid levels during fermentation decreased. Some of this could be due to adsorption and volatilization, but some of it is happening due to the fact that there's isomerization. In fact, when you look at hops, you don't really see a lot of beta-citronellol in hops. But during fermentation, you see beta-citronellol levels rise. And so what's happening is geraniol is being isomerized into beta-citronellol. Geraniol also can be isomerized into linalool, which in turn can be transformed into alpha-terpineol. And so beta-citronellol, geraniol can be seen as almost as like a pool of precursors for other compounds. And again, that makes the study of this both interesting and challenging, because it's a very dynamic interplay between these various components. And so it's hard, and I would caution you at looking at just one compound as the indicator of either hop aroma or evidence or lack thereof of a transformative event. Now let's talk about flavorless precursors that can be cleaved to release aromas. There's two classes of these. The first are the thiols. So an example here is a cysteine conjugate of a thiol. In this case, we've got the cysteine 4-MMP. So here's the 4-MMP thiol, this small sulfur-containing compound that has the parts per trillion odor detection threshold. When it's tethered to cysteine, it's odorless. And in fact, we find lots of this in hops. But in the presence of an enzyme that can cleave this, a cysteine beta lyase, can actually release the 4-MMP and result in the presence of this aroma. And we'll see some of these data later on in this presentation. Cysteine is not the only conjugate. We've got glutathione, and we've got cysteine glutathione, as well as, and these are not just also conjugated to 4-MMP, but also to 3-MH as well. So the idea here, odorless precursor that can be transformed in at least the odoriferous aroma compound can be released and start creating impact in beer. And the sister of this, or the cousin of this, is with the glycosides. So we can have glycosidically bound aroma precursors. And these glycosidic bonds occur with the terpene alcohols, so things like geraniol and linalool. An example here is a linalool glycoside. So here's the linalool molecule bound in this case to a sugar molecule, to glucose. And in the presence of beta-glucosidase, this bond is broken, and we release the linalool, and that sugar that is there. And there's some hypothesis that this may be yeast are doing this to try to scavenge sugar that's found in the precursors. Both the glycosidic rebound compounds and the cysteine conjugated compounds, we find these in different plant systems, in part because the compounds that are being bound to these sugars and amino acids themselves are hydrophobic, and the plant needs to be able to move them around within its system. And this is one way to do it. By tethering it to a sugar molecule or an amino acid, we can increase the polarity and allow the plant to move it around. So there is the potential that there's large portions of these precursors. And in fact, if you look at malt, we can actually see pretty high levels of thiol precursors that are there. And so there's some interesting possibilities there in terms of the use of beta lyase enzymes. Okay, one thing to point out is that this beta-glucosidase activity, and the same with beta lyase activity, varies depending upon yeast type and yeast strain. So we looked at this back to the work that Daniel Sharp, the guy who had spunk up the lab with the thiols. He was looking at beta-glucosidase activity in yeast. We looked at well over 50 yeast that came from our lab. Some came from Lallemand, some came from Kepler Streppen's lab. And what you should pay attention to is this dark blue dot here. This is the total beta-glucosidase activity. And notice that there's a pretty broad range here and within this subset of brewing yeast. And notice that there isn't really any order. We've got lager, ale, and Brett strains down here that are on the low end, and we got lager, ale, and Brett strains that are at high end. So it's while there's some work published early on that Brett seemed to have higher levels of this, we can find evidence where Brett has lower levels of this. So the point is that yeast strain varies in both beta-glucosidase activity and beta lyase activity. And so before we head out of this part of the talk, one thing to keep in mind is that there is an interplay here. So if we combine both the monotropian alcohol story, or isomerization story, and the beta-glucosidase story, you can see that maybe a hop has got a fair amount of geranyl glycoside. And if this geranyl glycoside gets hydrolyzed to release geranyl, do we see a bump in geranyl, or do we then see a further cascade of these compounds throughout fermentation or throughout dry hopping? So again, it's a very complex story here. So now let's move on to two case studies. This is a collaborative work we did last year with Lallemand. And the question here was, does yeast strain and the use of beta-glucosidase separately enhance or modify hoppy aroma or flavor in beer? And what we looked at were 4 different yeasts. Now, Nottingham in all the rest of these slides is basically our control. And they presented us with yeasts that are higher in beta-glucosidase activity, both the New England and BRY 97, and then an engineered Moscoma strain, which is even higher yet in beta-glucosidase activity. So the goal was to see whether these strains had an impact in terms of hop flavor with an eye towards the beta-glucosidase activity. And then separately, we looked at 2 enzymes. We looked at the ABV Aromazyme and then another enzyme we'll just call enzyme X. It's a third-party enzyme. And these are enzymes that are tailored for beta-glucosidase activity. So the story here is really around beta-glucosidase, but you'll see the results kind of move in a variety of different areas. Ester production, thiol release, so it's fun, interesting stuff. We made beers at the OSU Pilot Brewery. These are geared to be like an IPA. So it's not quite 15 plate OG hopping in the kettle, whirlpool, and dry hopping. We used Cascade, Centennial, and Amarillo. These were fermented individually with yeast, and in some cases with or without the beta-glucosidase enzymes. At the end of fermentation, post-dry hopping, post-BDK reduction, they were rough filtered, carbonated, stored in kegs before analysis. And the hopping regime on this was, I think it's relatively standard. 60-minute boil with a small hop addition on the front end. We didn't want these beers to be terribly bitter. A modest hop addition of Amarillo and Centennial in the whirlpool, and then a much larger addition during dry hopping. Dry hopping was occurring just at the very end of fermentation and lasted for 3 days before the hops and yeast were pushed out of the bottom of the fermenter. And then we wait for BDK reduction. We looked at the chemistry of these non-volatile components using alkalizer and bitterness units. We looked at volatile aromatics, terpene alcohols, esters, and we also thiols and thiol precursors. This was coming from Nicios, and that presentation that Tobias was talking about coming from Laurent, he's our contact at Nicios, so there's actually some interplay here as well. The beers were stored at 2 degrees Celsius and evaluated over different periods of time. We evaluated them within 1 to 3 weeks of fermentation, and then 10 weeks later, and then quite 15 or so weeks after, 15 to 20 weeks after we started fermentation. And the reason being is that these enzymes, whether they're enzymes coming from the yeast or whether they're enzymes that are exogenous enzymes that are being added, we're not inactivating them, and we're wondering, do we see changes that occur over time? And from a sensory perspective, we used a technique called projective mapping, and the data that I'm going to show you are really around a Catadata. That's check all that apply. So it's basically frequency data, frequency of use of specific attributes. And we've had 3 panels in these 3 different points in time, and this is the makeup of the panel. And I'll be showing you correspondence analysis of the Catadata. So let's dive into the yeast study. So here's the fermentation performance of the yeast, very much like slide 2 at the top. We're keen on looking at loss of extract and using that as a gauge of fermentation performance. The Nottingham fermented the fastest, the Moscoma yeast fermented the slowest, and there's a pretty broad range of fermentation speeds and a slight variation in terms of final attenuation levels. These values were, these results were to be expected based upon previous history with these yeast. I'm going to show you a handful of slides that are in this format for the next few set of slides. And basically the way it is, we've got different analytes at the top. And down here on the left-hand side, we've got Nottingham at the top, that's our control. And then we've got other treatments relative to Nottingham below it, and the actual values of the analyte in question. And then down below here, I'm just taking these analytes and expressing them as a percentage change relative to Nottingham. We think of the Nottingham yeast as a control. So what you can see here in terms of alcohol production, as well as the RDF, the real degree of fermentation. The BRY 97 had a higher degree of fermentability, as did the New England yeast, in terms of, and you see that also in terms of alcohol production relative to Nottingham. The Moscoma yeast had slightly similar levels, but lower levels of alcohol. A little bit of this is a challenge to completely interpret, because we get some change in fermentability when we dry hop because of the hop creep phenomenon. I find it interesting, there's a pretty broad range in terms of bitterness, and I don't yet quite know how to explain that. The treatments in these 3 yeast are at higher levels of bitterness. I don't know if that's a surface activity phenomena of the Nottingham stripping out more isos, who knows. The big question was, like, what happens to the hop compounds? And so what we can see is that it's not really a tremendously big change in linalool and nerol, but there are some relatively large differences in terms of geraniol and citronellol. We see higher levels of geraniol in the New England and BRI 97 relative to the Nottingham, and lower levels of beta-citronellol and lower levels of alpha-terpineol. So is this coming from the glycosides, or is this more isomerization, or a combination of both? It's hard to say, but there's clear differences in terms of the hop compound expression from the terpene alcohols. Turning our attention to the thiols, there's a pretty dramatic variation here as well. Notice that the Nottingham yeast was higher in the free thiols relative to the others, which I find kind of interesting to see. And similarly, the precursors in the other 3 are higher. So there's higher levels of precursors. Well, in the Nottingham yeast, lower levels of precursors, higher levels of free thiols. Keep in mind, though, that these precursor levels and the thiol levels span very different, they're very different magnitudes. We're talking microgram per liter in the thiols, precursors, and nanogram per liter in the free thiols. As you might expect, the ester and higher alcohol production varies, and that's kind of one of the key things that we know about yeast and have known for I would say for millennia, but certainly in the last century, looking at yeast biochemistry, is that they vary a lot in ester production. And so we see that the acetate esters tend to be higher in the Nottingham yeast, the ethyl esters are certainly higher in the Moscoma yeast. And there's very complex here. But I think one of the take-home points is that if we're going to be looking at things like thiols and things like these hop compounds, from a sensory perspective, we can't ignore the interplay with esters. And I think that there's going to be, we'll see interactions that will influence the perception of hop flavor. This diagram here is basically a way to wrap up those last 3 slides all in one package using a technique called principal component analysis. And the way to interpret this is that things that are close together are similar, things that are far apart are different. And so one of the big take-homes is that the Nottingham and New England yeast, from like a chemistry perspective, are sort of similar to each other and different from the Moscoma and the BRY97. And as we start looking in here, we can kind of see, okay, so the BRY97 had higher levels of geraniol, it had a higher RDF, it also had higher levels of the CG3MH precursor, while the Moscoma yeast tend to be higher in those ethyl esters, Nottingham higher in acetate esters, things that we're seeing that we saw before, but all in one package. And I kind of like this because it gives you a more holistic view of what's happening with the chemistry. Now let's turn our attention to the sensory aspect of this part of the study. So here are descriptors that came out of the Catadata analysis. And what we're seeing here is the percentage or the frequency with which these descriptors are being used. We're not actually scaling the intensity, but what we can infer is that if a descriptor is being used at a higher percentage, it's probably more important in describing that particular treatment, in this case yeast. And so we've got the 4 yeast treatments, and then within that we've got the 3 time points. And so you can see that the Moscoma yeast, while it was described as being citrusy initially, tended to have lower hits on a lot of these descriptors relative to Nottingham and certainly relative to the BR97. And you can see that things are changing with time. We can look at this kind of like the PCA, but using a correspondence analysis. And in this case, the red dots refer to the yeast treatments and the blue dots refer to the aromatic descriptors. There's too much here to see at once, so if we can kind of remove some of the data and look at things, like for instance, at that initial time point, this is a nice way to look at this. So you can see that both Nottingham and BRY97 were described as being tropical, stone fruit, resinous, sort of a little bit leaning towards this sort of DMS cannabis category, while the New England and the Moscoma were much more dominant in the citrus category relative to the tropical stone fruit. But the Moscoma also had onion garlic in it, as did some of the New England. And that's not unsurprising. If we think about like origins of hop aroma from the thiol perspective, we didn't talk at all about some of the sneaky compounds, but those OG aromas, those come from thiols as well. So if these are active in terms of thiol production, not only could we see some tropical, but we could also potentially see some OG coming from that. As these beers age, notice that we start to see movement from the right-hand side at 10 weeks, and then at 15 to 20 weeks, even further progression to the left-hand side. We're seeing a loss of citrus aroma, a slight loss of tropical, and a movement towards more of the sulfidic aromas. And this is something that we would expect to see. If you look at hoppy beer as it ages, it's not uncommon to see those bright tropical citrus aromas start to become muted. You see a production of aromas that are more like holiday fruit cake and sort of vegetative or compost or processed fruit kind of aromas. Looking at these individually, here are the 3 time points for Nottingham. It was interesting to see that Nottingham was relatively stable compared to, say, the New England, which really saw some pretty dynamic changes over time. Same thing with, not to a lesser degree with the BRY97, but also with the Moscoma yeast. So it's a complex story, but I think that the sensory element plays in nicely with the chemistry. I'm very much a fan of looking at sensory data because it gives you an idea of what's going on in a more holistic view. The take-home points from this part of the study, there were clear differences in fermentation rates. There was variation in hop compound by yeast strain. The New England and BRY97 were higher in geraniol. Nottingham was highest in citronella and alpha-tocophenyl. The Nottingham yeast produced higher levels of free thiols and lower levels of bound thiols. There's high variability with esters. And I think there's also distinct sensory differences in the yeast attributes with the Nottingham and BRY, starting similarly, more tropical resinous stone fruit relative to the New England and Moscoma, which were also tropical, but had some of these other chemical pungent aromas, the muscaroma being a little bit less aromatic. Now, I think a caveat on all this is that we just looked at an N of 1. We have not replicated the study, and so treat this as kind of like a concept trial. And again, do that with the next set of data as well. So last part of this is looking at enzymes. In this case, we had 2 enzymes relative to the enzyme-free, it's called enzyme-free, we're talking about exogenous enzymes of the Nottingham yeast. I noticed that there's some pretty significant differences in terms of final attenuation levels. We see that in the same set of data here. If you look at the ABV and RDF numbers, notice that this enzyme X, as well as the aromazyme, have higher fermentabilities. And I've seen this in other enzyme systems as well. You have to remember that industrial enzymes are not refined single enzyme acting on single substrate. They're more of a mixture. They often have side reactions, and it's not uncommon for an enzyme that is geared for carbohydrate glycoside hydrolysis to actually have effect on dextrin profiles as well. And so we see that fermentability goes up with these enzymes. We're seeing some side reactions in terms of dextrin reduction. We see some variation in terms of linalool, but I think it's interesting that actually these numbers are slightly lower relative to the control. The aspect, I mean, the response in the other hop compounds is relatively muted, certainly in comparison to the yeast effects. But that's not to say that there aren't sensory effects, which we will see to turn to the sensory data. These enzymes had some measurable effects on thiols and also thiol precursors. And so we see the ABV Aromazyme actually having higher levels of free thiol and slightly lower levels of the aroma precursors. So it could be that this enzyme, a side effect of this enzyme is that we're also seeing influence on thiols, even though the idea is that it's geared towards glycosides. Not as much excitement in the ester category, which you would sort of expect in this case, because the ester activity is coming from the yeast, not from these enzyme sources. And looking at this all wrapped up with the PCA, we see that the Nottingham control is sitting, in this case, down here in the lower south east quadrant, but the ABV Aromazyme and enzyme X are sitting in different quadrants. And the fact that these are all in 3 different quadrants means that these things are having an impact on the overall chemistry, different from what we'd see from the Nottingham control. So there is an influence on hop and fermentation chemistry. Now looking at the Catadata, we're not going to look at the biplots, but we'll just spend a little bit of time looking at the frequency data here. The setup is a little bit different here. We've got it by time as we go from left to right, and the sub-columns here are the different treatments. And what you'll see, like for instance, with enzyme X is that initially there's a fair amount of this OG aroma that is coming from this, and certainly it seemed to be higher in resinous characteristic. The ABV Aromazyme, it's got sort of similar levels of citrus and tropical fruit without the sort of the stinky aspects of this. But I think what's interesting, after a period of 10 weeks or so, notice that the descriptor frequency for the ABV Aromazyme is higher in a lot of these high-end descriptors, resinous, citrus, tropical fruit, and lower in herbal. So we're seeing something happening at that time. And then as we have extended aging, that those descriptor levels come back down to kind of what they were at the beginning of the experiment. One of the descriptors that we used in this was actually some scaling data. We asked panelists to identify whether these beers were either high, medium, or low in overall hop aroma. That's what OHI stands for, overall hop aroma intensity. And so you can see this is at the beginning of the study. You can see that the ABV Aromazyme had the highest level of high OHI and lower levels of the medium and lower level OHI. So there's some potential influence there that this enzyme is having in terms of hop aroma intensity expression. All right, so that's wrapping up my part of the talk. Actually, not yet. Just the overall conclusions here. We definitely see these influences of differences in fermentability because of side reactions on dextrin reduction by these enzymes. They had a modest impact on hop tripping alcohol. It was interesting to see that these enzymes did have some effect on thiol level release. And certainly the effect with esters is much more subtle. The ABV Aromazyme shows some potential. It shows potential for increasing hop aroma intensity. It shows potential for increasing resinous and citrus character with some aging. Of course, like you see with hoppy beer flavors, as the beers age, we start seeing this move towards more herbal. But it should point out that behavior seems to be more dynamic than Nottingham by itself. Okay, now the time for the wrap up here. And so I'm going to hand off to Joanne. Joanne's going to talk to you about some brewery case studies using the enzyme. And I see there's plenty of questions. I'll hop in there, try to answer some of those, and then we'll do some Q&A at the end of this as well. Joanne, you want to take it over?
Yes, thank you, Tom. Thank you for this very nice presentation. Yeah, it's always very nice to hear about biotransformation and all the science behind. So I'll move on to my presentation. Yep. All right. So I'm going to cover very briefly this part of the webinar with a talk based on the global trials we did in different breweries at global scale with this new product we just launched this week, which is ABV Aromazyme. Okay, so this will be more information for you in the practical sense, let's say. So this commercial product, it's an enzymatic preparation high in beta-glucosidase activity. And as Tom mentioned in his presentation, hop biotransformation is not just one single reaction, but it's multiple types of reactions happening. But in this case, Aromazyme is just covering one type of these explained reactions. In that case, the beta-glucosidase enzyme, which is able to release terpenes, okay, in that case, monotropian alcohols. So we did over 15 trials around the globe, as you can see in this slide here. You can see countries like Canada, France, United Kingdom, and I'm just gonna pick one of them, one of these trials, sorry, which was, I will say, one of the very first ones we did in this project, which was in Spain, thanks to the friends and colleagues of Napa Beer. So the guidelines the Napa Beer team followed in this trial is, as you can see here in these slides. So we asked them to produce one single batch, just to minimize variables in the process. So this batch was split into two fermentation vessels, as you can see here. Fermentation vessel 1, FB1, that was the control batch with no enzyme. And then the second part was put in another fermenter, FB2, fermentation vessel 2, in which the enzyme was added at the start of fermentation. Okay. In that trial we did at Napa Beer, we didn't hydrate the enzyme, although we recommend to hydrate for an even dispersion or an homogenization within the board matrix. And we know that some of our strains like Beer Wine 97 or New England have already higher beta-glucosidase activity compared to others like Nottingham. And as you can see here, we use Beer Wine 97 for that trial. The beer style that we brewed, it was a brut IPA. I have to admit here that this trial was done a couple of years ago because it was one of the first trials in the project. Brut IPA is, well, it's still there, but it's no longer a trend in the brewing industry now. We use 6 grams per hecto of ABV Aromazyme, as I said, at the start of fermentation. And as you can see here, some of the hops and quantities used in the process. So you can see 3 grams liter of Chinook and 3 grams liter of Centennial at the start of fermentation. Fermentation temperature was 18 C. And then the same amount of hops and hop varieties were added in the second dry hop at day 12 when fermentation was finished and the temperature was dropped down to 16. Following the same guidelines, one single batch was split into 2 different fermenters, and some samples from each tank or each fermentation vessel was sent to Mascoma which is a lab owned by Lallemand, where they did all the chemistry and all the analysis, as well as the sensory. So as you can see here in this slide, in this graph below, the red and blue bar, we checked for the total content of terpenes. Okay, so the control beer had this level of terpenes compared to the beer with the enzyme or treated with enzyme. So there is a clear increase of content of terpenes. And if we look at this table above, we can see here that linalool was the monotropian alcohol that was increased 3 times more by using this enzyme. This is not the same results we have seen in Tom's research, but it's important to note here that there are many variables, starting from the hop variety, the year crop, the point of addition of the hops, quantities, and so on. So every single thing counts here. But in this trial we did an upper beer, we just found this observation or these results here. Okay, that was the most significant one, 3 times more of linalool. So regarding the tasting, we had 2 different panels, 2 different tasting panels. One was a general, which was composed of people with no training on beer of flavors or beer sensory, whereas on the other hand we had an expert panel, also composed of 6 tasters, the same as the general panel. And the tasters, let's say, in this expert panel had a bit more training on beer sensory and of flavors. So as you can see in this graph here, the blue bar stands for the beer with the enzyme. So in the general panel, 4 of 6 tasters preferred the beer with enzyme. I forgot to mention here that both panels perform a blind tasting, so they didn't know what was in front of them. And on the other part, the expert panel, 5 out of 6 tasters prefer the beer with enzyme, again within a blind tasting. Okay, so that gives an idea which was preferred compared to the other. In that case, was the beer with Aromazyme, the enzyme. So in the expert panel, because they had a bit more of experience, we asked them about the descriptors and to score such descriptors. So as you can see here, in the Y axis, from 1 to 5, being 1 the lowest score, being 5 the highest score, tasters describe the beer with enzyme. As you can see here, we changed the color, now it's red. So the beer with enzyme was described as more hoppy compared to the control. Okay, by hoppy, here we don't describe or we don't differentiate if it's citrusy, floral, or herbal. It was just being more or less hoppy. Okay, another important or let's say main takeaway here, it was the bitterness. So beer treated with enzyme was a bit more bitter compared to the beer with no enzyme. We will assume here that because of the glucose release through the hydrolysis, so that will possibly make the beer slightly more drier. Okay, so that's why we recommend to do some adjustments before doing any commercial trial here. Okay. So after all these trials, we performed a global scale. They were really helpful for us to characterize this product. I saw before there was a question on that sense. So this product, this enzyme, is obtained from Aspergillus, a fungi culture. So this product is in powder form, and the stability, the shelf life of the product is about 4 years. So it's really stable. Okay, as already mentioned, is very high in beta-glucosidase activity. There is no side activity, for instance, in a melolytic activity. So it's not gonna eat the dextrins or possible astarage and so on. Okay, the recommended dosage is 5 grams per hecto. And the optimal activity, as you can see here in terms of pH and temperature, that makes this product suitable for a wide range of beer styles. In terms of pH, it goes from 3.5 to 6.5, being 3.5 low pH. That makes this product suitable for sour IPAs, sour ales, or why not even fruit beers. It's important to bear in mind that fruits also contain glycosides, so it's possible to release monotropian alcohols out from the fruits. Okay. On the temperature side, you can see here a wide range of temperature from 15 to 65. This will make the ABV Aromazyme suitable for bottom fermented beers, like let's say hoppy lagers, Indian pale lagers, which is IPL, as described here, or something more trendy nowadays, which is Quebec IPAs. Okay, for those who doesn't know, Quebec is a popular yeast strain from Scandinavia, which is widely used for New England IPAs or other sort of modern IPA beer styles. So it can be used also for this sort of beer styles. So some considerations, as I quickly mentioned before, it's important to do some trials in advance in order to optimize the recipe and define some process details. That means when to use the hops, at what point, which variety to use, the time of contact, and so on. Okay. And one takeaway from Laurent Dagan presentation that we discussed before with Tobias, and also it was mentioned in Tom's presentation. So Laurent Dagan stated that the use of hops in the whirlpool will mean or will impart some extraction of glycosides, which are precursors for this enzyme. Okay, so this will help also to release further terpenes in the fermentation process. So a brief summary of the listed benefits after all the trials performed. We can say that this product helps to increase the diversity of hop flavors and aroma by changing the variety of specific terpene compounds. That means that by using this product, you can release new terpenes like linalool or geraniol because they are bounded to sugar molecules in the form of glycosides. So it helps to change the profile, or let's say the terpene compounds in a hop. Okay. We also found that it enhances the beer mouthfeel and drinkability. And also we noticed that it reduces the unpleasant and harsh bitterness. We say here with brewers that when you have, for instance, a beer that it's been in contact a long time with hops, it has this sort of unpleasant bitterness, a bit harsh. So we just found that by using this enzyme, it can reduce this sensation. Okay. We also found that it increases the board fermentability, and that's mostly because of the glucose which is released from the glycoside compound. Okay, so you will expect at least 0.3 or 0.4 as average in percentage of ethanol increase. It's not much, but it's something to take into account. And last but not least, this product is really, really interesting to play with and to experiment, because it can express also a different character from less sophisticated hop varieties. We all know that in the brewing industry, there are certain varieties that are widely used or widely wanted, and sometimes it's difficult to find them. So you can use this product to play and change the terpene compounds of other sort of hop varieties. So it's really interesting to play and discover new things. So that was a brief summary from my side. I would like to thank you, Tom, for your very nice presentation and very, very exciting. Also, I would like to thank all the breweries involved in this project, especially Napa Beer. And if you have any question or any technical comment, you can shoot us an email at abvickers@lallemand.com, or you can visit us as well at our website lallemmandbrewing.com/aromazyme. Okay, if you have any more questions, we can discuss it right now. So thank you very much. Speak soon. Thank you.
Yeah. So thank you very much, Tom, and thank you very much, Joanne, as well. two very interesting presentations I found. And we also got quite a bit of questions here. And I saw that, Tom, you started answering them already.
Yeah, trying to get ahead of this as we're going to run out of time here.
Yeah,
so.
Exactly. We have a bit limited.
But we can certainly go into some of those that I've already answered. So
yes. Okay. So I think the one with the most vote was does pressure during fermentation influence biotransformation? So Tom, you gave an answer. Maybe you want to just.
Yeah. No. I mean, as I point out, I think it's evident that pressure certainly has an influence on ester production. We know that, and brewers use that to their advantage or in some cases disadvantage, depending upon how as they scale up in terms of fermentation size. But I can't speculate right now as to whether that will have an influence on biotransformation. It's certainly going to have an influence on hoppy expression because of interplay between yeast esters and the hop compounds. But whether it actually slows down or accelerates things like glycosidase activity or cysteine hydrolysis or esterase activity, no, I don't know. Maybe it has an influence there in terms of the modification of drain ale. So it's a great question, an area that we should dig in further.
Yeah, and well, I can say we haven't done any pressurized fermentations either, so unfortunately I can't. Or Joanne, I think we can't chip in here.
Yeah.
So next question was, can you control the concentration of the 4-MMP during fermentation to have that more tropical rye B character versus a cat P?
Yeah, that's a challenge. I think one of the things that as I mentioned, that answer that we see is that in the lab here is that beers that when we first brew them, they can be really stinky. If we're using really thiol-rich hops, they can move into that area where they're almost offensive. And then with time, that thing goes away. In fact, we had a beer maybe 4 or 5 years ago that we brewed that when we first smelled it, it smelled like peaches and armpits. So it had stone fruit and really sweaty quality. And 6 weeks later, we renamed it to Slice of Heaven because that armpit quality disappeared, and it was kind of like this mixture of stone fruit and tropical, and it was just this delicious smelling beer. Granted, this is a research lab, not a production brewery, but I think it's generally accepted that the hop flavor is pretty dynamic, particularly right after fermentation, and certainly this thiol aspect of that there's a rapid change between beers that can be extremely resinous, but also extremely stinky right when they come out of dry hopping to post-aging effects.
Good. Whoops.
I didn't really get at answering how do you actually control it. I don't know how what lever you can pull. You can certainly by pulling hard on the levers of hops that are really high in terms of thiol, or even selecting hops. At hop selection that kind of move in that really stinky category, that's certainly going to accentuate it in beer. But in terms of dialing it from a yeast perspective, I don't have a sense right now.
Yeah, and again, we don't have an answer there as well. We're just at the beginning, basically, of this whole interaction of yeast and hop, and hopefully we're doing a bit more projects on that next year. So maybe next year we can give you more answers on that. So the next question is coming from Sam from Pressure Drop in the UK. Thanks for the presentation. Awesome. In the Napa beer study, fermentation occurs at 18 degrees Celsius with a second dry hop at 16 degrees Celsius. Given that the beta-glucosidase optimum temperature is 45 to 55 degrees Celsius, is it possible to ensure the full level of enzyme activity and full attenuation in the fermentation vessel at 16 to 20 degrees Celsius? Joanne?
Yeah, well, that's a really good question. Thank you, Sam. Well, as we have discussed, the optimal temperature can go up to 65. So that will mean that the enzyme, I will say, the full activity is done in few hours. If you go down in temperature, it will need more time. So you don't need to go at that level of temperature, let's say 45 or 55 to reach full activity. You have enough days during fermentation to allow the enzyme to do the job. So that's why we recommend to use it at the start of fermentation to have enough time. Just to give you an example, we have another enzyme at AB Vickers, which is a glucoamylase. Glucoamylase can work in just 1 hour or hour and a half at 65 Celsius. Okay, but if you use it in the fermenter, which is at much lower temperature, you will have a full degree of attenuation by using this enzyme because of the time, not the temperature, but the time. So I hope I reply your question.
Yeah,
Sam.
hey, Joan, you even look further down, Sam also kind of follows up on that, asking about if you're going to use this enzyme with the Kvike yeast fermentation at a much higher temperature, like, but that might actually promote activity. It's an interesting thought.
Yeah.
It probably will, I would imagine, because so that's a good thing.
We should take into account as well that higher temperatures will mean possibly more CO2 stripping, but everything has to be taken into account here.
Good. The next question, what about the use of years-old hops lost their bittering? How are they affected by transformation and aromazyme?
I don't know, is the hop industry going to love this or hate this? Go find your old hops and biotransform all those medium chain stinky fatty acids into something aromatic. I would say that it's not without possibility that using this stinky hops is going to result in a pretty wonderful smelling beer. Now, it just depends of the timing of their addition and the yeast esterase activity. I would think that if you're going to dry hop with these and you don't have high levels of yeast in suspension, that you're still going to have kind of a stinky beer. But if used further upstream in the process, I don't see why not. You look at Lallemand brewers that they use old hops. They use old hops, I think, for a number of reasons, like they're cheap, and brewers can generally be people that are really focused on the bottom line, but they also have low ISO potential because of all the hop oxidation. When you brew with those hops, you don't get high levels of isoalpha acid, so you can get the bacteriostatic effect of the yeast without having tremendous impact of bitterness. And some of those yeast over time create really delicious flavors. Now that sort of smelly medium chain fatty acid stuff happens much later on. If you smell a lambic fermentation after 8 months or 12 months, it kind of goes to this phase where the yeast are actually creating a lot of that stink. And I think a lot of that, more that's coming from the yeast than from the hops. Long-winded answer. I think that there is the potential of creating nice aromas out of stinky hops. That's to be determined.
Good. Next question. We have an in-house yeast. Are there techniques to analyze yeast for biotransforming mechanisms? Relatively straightforward. Tom, you mentioned you screened all.
Yeah, so there's a number of different approaches to doing this, and I would say they have a modest level of complexity. They're not highly complex, using really highly sophisticated instrumentation, but they're more complex than just being able to do on the benchtop in a brewery. So a common one is to grow a yeast on a substrate-specific material. So that could be with a glycoside, like an octyl glycoside, and just see whether the yeast can actually hydrolyze that and grow on it. The slide that I showed, we used a fluoroprobe, this 4-mug. So again, another mouthful. It's like 4-methyl something glycopyranoside. But basically, it's the same kind of idea. You got a glucose molecule tethered to this fluoroprobe, and as the yeast ferment it or react with it, they hydrolyze the fluoroprobe, and then the fluoroprobe can fluoresce. And in a fluorescent spectrometer, you can actually see it. But that then requires having a spectrometer that can deal with fluorescent emission and then handling those reagents. So I don't know, in your labs, you guys have characterized this.
Yeah, we have, as I said, characterized our strains, and we used a similar technique. So we have also these selective media, in particular for the beta lyase activity. There's actually a paper from Belda, I believe, from the wine industry.
Yeah.
Where we base that on. It's S-methyl L-cysteine media.
Yep.
With,
so one of the challenges that we found is that when we worked with a specific probe like this, like those data that I showed you in the slide, we can see a pretty broad characterization of differences in activity. But when we transmit that into like a fermentation, And we add another probe. In this case, we'll use an octylglycoside. And so we can look during fermentation. We see the release of octanol. I can find octanol coming from yeast or fermentation. So we know it's coming from this probe. We get release, but the extent of release was not as big as we thought. And we didn't see as much differentiation in the release of that probe as we did see in the fluoroprobe assay. And so it's sort of unsatisfied in some regard that you can see with one method a big set of variation, but with another method or more like a brewing application to see not as extensive a variation. If there were a really big big lever, you would see people pulling on it, right? Like if a particular yeast was really having a dramatic impact, then kind of the silver bullet, then people would be using it. It doesn't mean that there aren't things in the future with genetically modified yeast that might be engineered for high glycosidase or high beta lyase activity that we can really crank that expression up. Yep. Right now, if it's there, it's functioning, but it's not the silver bullet, shall I say.
Yeah. No, I mean, we had also different techniques, and sometimes you get, as you said, a wider range of results, or we don't see a huge difference. And there's also some genetic tests that you could do. So I don't know if that's benchmark, but if you have a machine's PCR basically for that, you could look at gene expression and so on.
And now you're into really complicated approaches to trying to characterize that yeast.
As I said, sometimes they're a bit contradicting, but if you pull them all together, you get an idea which strain has a higher activity or lower activity. Good. The next question, I think we kind of answered that already. That was regarding the quake. If you could use quake and basically give with a higher temperature of a quake fermentation, get a higher enzymatic activity. The next one is more details on aroma masking, please. So,
okay, yeah, I went right by that really quickly. Yeah, so the concept with aroma masking is that the presence of one aroma influences your perception of another aroma compound. And in some cases, you can have just such a high level of one compound that you can't smell the other. Think of it like with sound. Like if you've got the radio or someone on the phone, and if the TV is on really loud and you can't actually hear what's on the phone, then okay, that's when this is getting in the way, but that's masking. The masking part is actually one compound interfering with your ability to sense or diminishing your ability to sense another compound. And we have not done a lot of work in the masking area. But the thing I mentioned about POF positive results was just that it came from a concept trial with Daniel Sharp's research where we were looking at a range of different yeasts. We had one yeast that we knew was high in beta glucosidase activity. It was also POF positive. And when we made beers with that, it ended up being a wine strain. The beer didn't fully attenuate, and the beer had intense levels of 4VG. And we found that the sensory panels scores for the hop aroma was lower with that hop, with that particular yeast. And of course, the 4VG and also the isoamyl acetate banana levels were off the chart. So I don't know if it's conclusive that that's masking, but certainly it was sort of interference. I've had some delicious beers that are hoppy and POF positive. So I'm not saying that that's POF positive is a bad thing for hops, but I think that it's hard to look at and one particular compound or even a particular slice of aromatic descriptors and in the absence of the others. Right? So things like POF positive or things that are really high, I might just intuit or suspect that if you have really high levels of esterase activity, things that produce these more red fruit, banana, that kind of stuff, that has the potential to actually accentuate some hop aroma as well. So.
Thank you. The next one. Have you tried adding the ABV Aromazyme to finished beers, for instance, in the keg or post-fermented samples? How many days, hours does it take?
Right, exactly. You guys want to? We haven't done that. Have you?
Yeah. Joanne, you answered. You're muted.
You're muted. Still muted.
Sorry.
Yeah.
I was replying by saying that because of this hydrolysis reaction, some glucose molecules will be released and then risk of overcarbonation. So we don't recommend to use that one at the end of fermentation. Yeah.
Then I think that's the second last. Glycosidic cleavage leads to glucose production. Is this glucose metabolized by yeast and increases ethanol concentration? If so, what extent does it actually matter?
Yeah. In theory, it should, but in fact, some of the initial work that was done on glycosides by Pat Ting at Miller Coors, they used some C14 labeled glycosides. They could actually see, like, during lagering, the production of these compounds, and the hypothesis there is that the yeast are looking for a carbon source. But keep in mind that the level of these precursors are relatively small in hops. So we're talking in the range of in the microgram per liter level, not milligram per liter. And even if you're at milligram per liter levels, to see an effect on ethanol, we need to be talking about like 10,000 milligrams per liter, right? You need to be in the percent level or tenth of percent level in order to see an influence. So I would not be worried about this impacting the glycoside impacting ethanol getting out of spec. That being said, the use of the enzyme, like the ABV enzyme or like that enzyme X or any enzyme that people sell you that say has glucosidase activity, keep in mind that could actually change the fermentability of the wort, and so what you think you're going to hit in terms of finished apparent extract may actually be lower, and hence your alcohols may be higher. So.
Yep. Good. Next question. Color. Appreciate the discussion of the sensory impact on aroma and taste. Has much work been done on the impact on fruit glycosides responsible for color? I'm thinking of what I've observed in fruit beers. I don't have.
No, personally, I wouldn't think that the glycosides would have an impact on color. The color part that you see from like a plum or a grape, that's coming from an anthocyanin, not from a glucosidase. Some of those do have some glucose moieties on them, some of these polyphenols, but no, I don't see, when we look at the glycosidic fraction, well, when we make an extract of hops, a water-soluble extract of hops, there's a little bit of color, but it's not, I don't see that being a driver of color flavor or even a modifier of color flavor, it's of color intensity.
Good. Next question. Let's move. Are there data on beta-glucosidase genetic variation across the yeast discussed? For instance, different genotypes, different isozymes with different levels of activity, gene copy number, and/or gene expression levels, which correlates with the results Tom presented. And
yeah. Okay. So I'm sure there are. The samples that we got from Kevin Verstreppen's lab, he knows the background on those, or at least some of them. I'm not sure how much that he's characterized, and I don't know the degree to which the ones that the Lallemand lab sent me have been characterized. But we haven't ourselves connect the dots. I think I know that Kevin is trying to do it, and other labs are trying to engineer strains. This is kind of what Moscoma is doing, essentially, is trying to look at the genetics of glycosidase expression as well as beta lyase expression. So I don't know. Tobias, do you want to?
Yeah, I mean, we have, as I said, we have this genetically engineered yeast that is really high in beta-glucosidase. Other than that, we haven't screened really our yeasts genetically for beta-glucosidase. We have done that for beta lyase, so we have some results for that, but not on beta-glucosidase yet. Good. Next, oops, I'm moving too fast. First, is there any way you can promote the formation of new glycosides during hot side brewing in order to protect the aroma until a putative exogenous enzyme is added, maybe when fermentation is ending? Second, how stable are the secreted enzymes from bread? For example, in 1 year bread beer could have active enzymes of its final beer to interact with a fresh, highly hopped beer.
Oh, good questions. Yeah.
Yeah.
So certainly, if the idea is that the flavor component that's coming from the glycosides is going to be important enough in terms of the overall flavor of the beer that's being produced, finding ways to get more glycosides in solution is like a key there. And so, in that case, maybe a late whirlpool addition might help. I mean, even a boiling addition will ensure greater extraction, most likely, than a 2 or 3 day dry hop addition. And so that was kind of a theory that people have postulated. If you have a sample that is low in free geraniol but high in free geraniol glycoside, that you might use those hops, the high geraniol glycoside hops, in the kettle or in late hopping. You're stuck on the kettle with hop isomerization. So if you have a hop that's got a lot of alpha acid, you're limited as to how much you're going to put in the kettle because you're going to get a lot of bitterness. But a late hop addition, where you can get more potential extraction because you have heat, may be a possibility. But that being said, we haven't actually tracked the extent of glycoside transfer from hops into wort or from hops into beer, in part because they're difficult to measure in both hops and beer.
Thank you.
Oh, and then the second part was like, do these enzymes, right? Yeah, after a year? Oh, that's a good question, right? So presumably they do. I mean, certainly the Brettanomyces is growing along quite happily after 8 to 12 months of fermentation, and let's say alembic fermentation. But does the enzyme excreted by, let's say, to take that Muscoma yeast that's got high glucosidase activity, so if it excretes some of this enzyme, does it persist a year or two later? I don't know. My intuition would say no, because I don't think that those enzymes are going to be that stable, and there's going to be some interaction. But I don't. Do you know, in working with other systems, wine systems, or whatnot? Totally do.
No, I mean, we haven't done any long-term studies in this either.
Yeah.
So I couldn't really tell.
Sure.
Joanne, yeah, no. No. Then we have the next question. Do Lallemand plan to add Aromazyme in some of the 10-gram yeast packs so it's combined? Any plans? Might be a good idea, but as far as I know, there's no plan to do that in any of the as a combined enzyme yeast pack yet. Any info on carryover and serial repitching situations? Can an enzyme overdose situation develop across time? I don't.
Oh, you're talking about, like, I'm not sure if this is coming from the use of the ABV Aromazyme, and probably, then that happening. I don't see an overdose happening on just like the yeast expression side, but in terms of an exogenous enzyme.
Yeah, I'm sorry, we have to speed up a little bit. We're quite over time.
Yep.
So we have maybe one or two more. Thank you very much for this presentation. I have a question regarding this enzyme. In other words, enzyme in general. I don't know much about, but I know they work using cofactors as a main fuel. Regarding Aromazyme, does it already contain some cofactor inside, or is it too? Or does yeast produce it?
It's a hard question to ask. Yeah, certainly enzymes need cofactors. They need things like they're stabilized by zinc or stabilized by metal ions. Running under the assumption that there's enough of this already within the yeast itself, that's not a limiting aspect here.
Yeah, and we don't add any cofactors. It's a pure enzyme that we provide.
Can you see the question that there's one that's like sour Philly? Sour Philly is sitting at the top of mine with 5 votes.
Yeah, on the top.
Oh, sorry. Yeah, I missed that one. Sour Philly can work to pH of 3.2. How would Bio-T be affected by such low
pH? Yeah, I think there was someone that had asked a question about the influence of pH on this. I would certainly think that pH is going to have an influence on the extent of biotransformation. And I've heard anecdotally some brewers playing around with sour worts and trying to modify hop expressions. But other than saying, I think it's going to have an impact, I can't tell you which way or the other. But yeah, I mean, a sour Philly is a sour producing Saccharomyces, correct? That Lallemand?
It's not a Saccharomyces.
Okay, not Saccharomyces. Yeah, it's an environmental strain, right? That came in Pennsylvania.
La Chance, actually.
Yeah.
Subspecies. Good. We still have lots of questions. I don't know. Maybe we can do one more, and then we have to wrap up. And we probably can see these questions and then answer by email also. Last one then. How would Aromazyme work in case of lagers fermenting with yeast, like the diamond, at temperatures of 8 degrees Celsius or lower? does such a low temperature render it inactive, or it takes even more time to notice its benefits? Joanne, do you...
Yeah, well, we we already slightly replied this question before. You can use it at low temperatures. It will just go slower, that's all. But it's not going to be ineffective or just a stop. Yeah, so no problem on that.
Mm-hmm. Yeah, I would agree. I think it's worth exploration on a lager situation. You think about enzymes that are being used, enzymes that are very temperature, have high temperature tolerance and high temperature activity, like AMG. You see those being used during lager ferments, and they work just fine. They'll dry out a beer in no time. So I would think that while temperature is going to slow things down, it's not going to stop it in this case.
Mm-hmm. I just saw that someone re-entered the question. Apparently we skipped over that, so I'm going to do this and then we finish. This question was good. So on a homebrewing level, what would you say are most important factors to get the most out of your hops? Water composition, fermentation conditions, temperature, addition times. Any recommendations?
Let's see in this question. Joanne, do you want to answer that? I didn't.
Yeah, sorry, I just missed the question because I'm looking.
Oh, sorry. Okay, so on a homebrewing level, what would you say are most important factors to get the most out of your hops? Water composition, fermentation conditions, temperature, addition times.
Everything.
Yeah, well, yeah. I would think that addition time, hop selection, hop amount, those are the big drivers. I think water. Certainly, water has an influence on flavor, but in terms of hop aroma expression, I think it's lower. It's like a tertiary influence. I think malt will have a bigger influence in that, in terms of the overall perception that you get in the finished beer. So, but certainly timing of when you add hops, the hops that you choose to use, and certainly the amount are going to have the biggest drivers in terms of hop expression.
Mm-hmm. Good. There's still plenty more very interesting questions, but unfortunately we're running out of time. So I want to thank you again, Tom, and also Joanne.
Yeah,
very
nice
presentation.
Sharing some research with you and the rest of the brewing community.
Good. And thank you very much, everybody, for listening and also for these great questions. And I think we can answer some of them offline then. We do that. So thank you very much, everybody, and goodbye until the next time then.
Thank you.